menu things. If you
Yeah, you try it.
Oh my gosh, these are like,
Oh,
they call that, there you go. We're gonna be stuck with the learn bar.
yeah, I think we decided that last time.
Right. But just do an audio check for folks online. Can you do like a thumbs up or confirm you're able to hear us?
Hear you.
Okay, great. Thank you. All right. Light attendance in the room, but glad we have the hybrid connection for folks. So we've gone through a lot of these slides, information before, so some of this may be really familiar to a lot of folks here, but want to make sure we have a good similar message for folks who haven't heard it, and so we'll make this recording available online. So we'll just get into it here. So my name is Keith Hannon. I'm with the Army Corps of Engineers, New England District. I'm the project manager for the Connecticut River Hydrilla Research and Demonstration Project. And on the line is Dr. Ben Sperry. He's with the Corps' research group, the the Engineering Research and Development Center, ERDIC. And he's heading up the research project. Slide. So we'll go through here. So really, just quick summary of what we'll do with these with the slides and the information we're presenting. So we'll give a little background on hydrilla, the invasion of hydrilla, and what it's doing in the Connecticut River, and just so people understand a little bit more about the basics about the plant and what we know about it. We'll get into more details about the Corps' current demonstration project and the research we're doing. And then, of course, we'll open up for discussion and questions. And really the ultimate goal is just for the public to understand the details of the Corps project and all the concerning issues about the plant. But this is a really important meeting to do this here now because we are moving from Connecticut into Massachusetts. So the last couple of years, we've been only focused in Connecticut. So this summer, we are crossing the state boundary into Massachusetts. And we'll get into more of those details as the presentation proceeds in a bit. So the origin story of this Connecticut River Hydrilla started around 2016 when this unusual plant was found in Keeney Cove. It had just really interesting growth characteristics. There was just a lot of plant matter and biomass that really filled in the water column in a way that people who are studying these sorts of plants were not familiar. And so it was discovered during a BioBlitz in 2016. And this plant was then genetically tested and it was confirmed to be a unique strain of this plant. It's genetically distinct plant. So it had a first time being observed in the area. So there's a number of project partners that we're working with. Margot Burns is with the Lower Connecticut River Valley Council of Governments. Greg Bugby in his office at the Connecticut Ag Experiment Station. And then Mass DCR, we're working very closely with Kara Slowski and Jim Straub, and Kara's here in the room with us this evening. And additional stakeholders, mostly Connecticut-based at this point. I won't go through reading all of them, and then additional research partners that have helped with this project along the way so far. And then, of course, the government agencies we're interfacing with and coordinating with. So the Connecticut River is a very long, over 400 miles long, really important natural resource in the area. It's an American Heritage River and the first national blueway, congressionally mandated blueway. So it's obviously an important river system. And so when this plant appears in it, it gets a lot of attention. And so we'll get into more of the details there. So in general, this is a timeline starting around 2018 when the genetic testing was conducted and it was confirmed this was a unique strain of the plant. But then through 2019 through 2021, there's a series of surveys that were done in the river by the Connecticut Ag Experiment Station. Won't get into too much of what's all these details here, but this is a good summary. And then leading up to 2023, there was a lot of work leading up to the Corps getting involved in 2023. The River Cog and Margo were very instrumental in getting stakeholder and state center support from many of the New England states to get federal funding to start this project with the Corps of Engineers. So we successfully kicked off the project in 23, and we had a successful implementation last year in Connecticut where we'll get into some of those details more, but we were able to do field demonstrations to show how to control hydrilla last summer. So we're looking to continue that work through this project. And so, right, so this is 2025. We're looking to expand into Massachusetts. So Mass DCR is our close partner in that work. And Kara will talk a little bit more in a bit about this, our partnership for this project. Wanted to give a shout out to the Coast Guard. In 2018, they helped conduct some of the first surveys to start tracking this new plant. And so for the folks who have been observing this plant, those stakeholders are been negatively impacted by its the growth and density of this plant. So they're seeking an aggressive control and management response to this invasion. The Connecticut River, like I mentioned earlier, is really important environmental resource and also very important for the local economy. There's no official eradication or control program in the state of Connecticut. You know, there's a grant program available, money is available to help folks to combat and try to control hydrilla, but there's no official state-led effort at this point. So we're hoping to contribute to that and to develop best management practices to help the local entities and the states deal with this. So the Corps specifically has been involved with studying hydrilla since the 1960s. The Corps' Aquatic Plant Control Research Program has been leading that, and that's who is involved with this project now. And the Corps of Engineers, to do this research project, we work under the Section 104 of the River and Harbors Act of 1958. That's the authority. And so in fiscal year 23, we received $6 million to kick this study off. And then again, the following year, we received $5 million. But due to the current continuing resolution that we're under, the program did not receive any new funding. So we'll talk about what the effects of that has been on the project so far, but we are planning on doing some work this summer, but it's going to be dialed back. The level of effort is not going to be as much as we had planned. So this was just signed letter from multiple New England senators, basically recommending that the appropriation for this Connecticut River Hydrilla focused research project receive appropriations. So that's what led to that first, 2023, appropriation that we received. So overall, the project objectives for the Corps' demonstration project is to implement science that protects public and environmental safety, to conduct research to develop and evaluate best management practices, and then to develop guidelines and recommendations and do a technology transfer of investment management practices to the local entities and the states to implement the results that the Corps was able to solidify and for recommendations. So the overall goal for this team and our work is to protect and restore the Connecticut River, its tributaries, and the associated ecological and economic benefits from hydrilla invasion impacts. So in part, doing that, some elements that we're also engaged in or include public engagement. That's what these public meetings are being run for. We want to help educate and inform the local groups and the states as much as possible and to coordinate. We're also doing environmental field studies, data collection, to inform different treatment plans and recommendations. We're doing federal and state permitting. And then ultimately we wanted to demonstrate treatment with herbicides and then monitor those results and publish the results. And so this is just a general timeline of, phase one, we do these field efforts, data collection, and that informs phase two, the implementation, which is to demonstrate the treatment. And that's what we did last summer, was the first round of demonstrating treatment. So next slide. So first and foremost, the public safety is the primary goal for the Corps on this project. When we considered multiple options on how best to control hydrilla, but we also are very mindful of the safety when using potential use of EPA-approved aquatic herbicides. So we're looking to develop strategy and a treatment plan that uses less and saves money. So, and we want to really remove as much of the hydrilla at the selected treatment sites as possible. And to restore, ultimately to show that we can, this is a good method to restore the Connecticut River and its tributaries and return the associated ecological and economic benefits that may have been negatively impacted. So we'll get into some of the background of hydrilla and how it arrived and spread through the U.S. So it's not native to North America. It arrived in Florida around 1950s from a tropical fish aquarium dealer who dumped some of this plant into a canal, and that started the first infestation and spread of this plant down south in Florida. A second strain was found in the 80s in the Potomac River, and then we're now around 2020 was that the new strain that you're talking about, the Connecticut River strain, was discovered in the lower Connecticut River. So there's now 3 strains to be aware of. And so this was a screenshot of the larger data set from the surveys that were done by the Connecticut Ag Experiment Station, starting in this area in the Agawam, Long Meadow, going all the way down the river. You can see all those dots are confirmed locations of where hydrilla is growing, this new strain. So you get a sense of how expansive it's become. And then in addition to the direct effects to the river, there is an increasing concern that the plant is leaving the river and appearing in isolated lakes and ponds. And so that's being shown here in this map. And And the red stars are the Connecticut strain of hydrilla that has jumped pretty far from the river itself. So including the northwest corner and East Twin Lake, that's pretty far away from the river. So it is finding ways to spread and be carried from its source in the river to these isolated locations. So that has a lot of people concerned. So in 2023, there was 6 confirmed isolated lakes or ponds. And then last year in 2024 is up to 10. So it seems to be increasing. So some of the differences between the different strains, there's the monoecious plant, the strain of hydrilla, where the male and female parts are on one plant. And there's also the dioecious strain where they're on different plants. The male and female plants are on or parts are on different plants. And then the new strain, the northern hydrilla in the Connecticut River, is looking like it's more of a monoecious strain, but some of these similarities, like having these tubers, that are still kind of being discovered. And so Ben will talk a little bit more about what that's looking like now, but it was a little confusing for folks at the beginning. There was no parent tuber, which is very common with the other strains of hydrilla. And so you can start to see in the southern US in the orange, that's the dioecious strain. And then the more of the green color is more of the monoecious strain. And that's been in the Northeast since about the 80s. And you can't really see it that well, but in Connecticut, there's a new polygon there showing a slightly different color, and that's the Connecticut River strain. It's a new strain in the Northeast. And then this map on the right is just showing with climate change the projections of where this hydrilla is likely to be growing successfully in a warming climate. So one of the reasons hydrilla is such a concern is that it spreads very well and very quickly. So how does it do that? So it grows very rapidly in the summer from these propagules that form on the stem. Those are called turions. Those break off and can disperse. They fall to the bottom where they find sediment, and then they can sprout, or they lay dormant till the following spring, and they'll start to grow roots and grow new plants. So in addition, if the plant gets cut by, say, like a boat or something like that, the fragments will float and disperse as well, and those can also generate new plants. And so the wind and the currents, birds, it's a piece of this plant gets wrapped around its body, it can transport it to a new location, or if there's fragments wrapped around any part of a boat or a boat trailer, that also can transport this plant to new water bodies. But we're also finding that this Connecticut River strain is extremely prolific with its formations and productions of turions, and Ben will get into more of those details in a bit. So I thought this was an interesting slide. It's in Connecticut. This is Hamburg Cove, but it just kind of shows how the plant has spread from 2019 to 2024. You can get a sense for just the area has it's spread and the density has increased over that period. So just to get a sense of, how it's spreading. Go ahead. So this strain of hydrilla, hydrilla in general, it's a perennial plant. So it grows in very dense mats because it grows so quickly and efficiently. Hydrilla in general can grow over 25 feet tall under the right conditions, so it can fill an entire water column that's that deep. And so when it grows and hits the surface, it can also start to grow, spread laterally. So there's just an awful lot of biomass that can fill in the water. So it's problematic for a lot of the native plants. It blocks light and absorbs a lot of the dissolved oxygen, which can be, have an effect on aquatic wildlife like fish. It is somewhat salt tolerant, but it also is adapted to grow in very low light and low CO2 conditions. And so generally it can double in biomass every 2 weeks during the growing season. And so it's also referred to as the perfect weed because it tolerates a wide range of water conditions. It easily reproduces, spreads by multiple methods, and is difficult to control. This was based off of a study that was done at the University of Florida. They were able to grow this plant and track its growth rate. So after 5 weeks, they demonstrated that it had grown 16 feet. So that was averaging about 3 inches a day. And so this is not the Connecticut River strain of hydrilla, which may actually grow faster than this. And then the other analysis on the plant is, it branches. Every new stem will branch, and then that new branch will branch. And so each new stem or branch of this plant will continue to grow at that approximately 3 inches a day. So that's how you start to understand how quickly this plant spreads and fills the water column with so much biomass. And you compare its growth or number of branches produced to a native pondweed. It's orders of magnitude different. And so this was taken in the Connecticut River last year at Agawam, looking south. You can see Six Flags there in the distance. So this green plant in the water there. That's all hydrilla. So this is where we're studying it in Massachusetts. Some other representative photos. This was a drone image collected by the Connecticut Ag Experiment Station at the Mattabesset River in Middletown, and you can see what looks kind of like a tidal mudflat is actually on the edges of this open channel. That's all hydrilla. And so the open channel in the middle is really likely just kept open from possibly just occasional boat traffic. But it had that channel not been there, this whole river would have been completely filled in with the plant, and it just goes off into the distance. This was taken at Chester Boat Basin. See a representative shot of it growing in some of these marinas and boat basins in lower Connecticut River. And then this was at Selden Cove during pre-treatment. We actually got a little stuck there. We had to get the prop unfouled because it just wraps around the propellers, but you can see how much hydrilla was filling the water there, and it goes all the way from the surface to the bottom, and that was probably about 10 feet of water right there. And then this is at Portland Boatworks, again, interfering, making it very difficult for boats to access these docks. And then in the northern extent that we've been able to confirm as of last year of the plant in the river is in Agawam. And so that map is just demonstrating that anything south of there, you just see the hydrilla is mapped. And this is a quick underwater video for those who haven't seen this, just to get a sense of what hydrilla looks like underwater, because we show a lot of photos of what it looks like above the water. But we just put a GoPro camera underneath, and this was from September 2023. And there's some areas where light gets through and some openings, but you'll notice it just gets, the light does get blocked out pretty well in the areas that are not in these openings. So a lot of these, there's a couple of these videos, they're on our the project website, and they're uploaded to YouTube, so you can check those out too another time. Click outside the There you go. Okay, so we'll get into, the impacts the hydrilla is, having to recreation, the economy, and to the, ecological impacts. Next slide. So I mentioned how, the plant does wrap around the props of propellers on boats, and it becomes a real problem. You have to, you can't boat through it when that happens. So there's a lot of concerns with, the plant compromising, marina and boat basin, being able to function during their really important time of year, midsummer. There's concerns about, general concerns about fishing and recreation loss associated with boating and swimming and those sorts of activities. It makes it very hard to do any of those things. Impacts to the economy are linked to oftentimes the recreational loss, but they're just the general compromised navigation abilities in the river and the back basins and tributaries. It can be a real problem. Like I mentioned, accessing boat basins and docks affects the livelihoods of many people. It can have a negative effect on the tourism. There's some numbers provided on what the lifestyle economy and what the value is in the Connecticut River. That was provided to us from River Cog. So in general, yeah, there's just a lot of, well, there's multiple industries that rely on a healthy, functioning river system. So that's being compromised with this plant. And something else to just note is because the plant fills so much of the water column, it actually is, there's concerns that can contribute to increased flood risk. It doesn't allow for the efficient flow of water downstream. It might start pushing it laterally. So that's another consideration that we want to address. The ecological impacts are significant. It just grows into forms large monocultures, and so these large stands crowd out native species, aquatic plant species, and generally contributes to a loss of biodiversity. So there's just other concerns too that, based on research, which we'll show in a second, a lot of the invasive aquatic plants are they harbor cyanobacteria, but there's so much biomass and growth with hydrilla that the cyanobacteria risks are even higher than other aquatic invasives, and so that's been studied, and the effects of that has been a concern. So yeah, next slide. And this is a good example of a native plant of freshwater eelgrass, and the Connecticut River strain of hydrilla is growing right next to it. So this sort of interface right now is of great concern. Species like eelgrass are really important for river herring or the Manhattan fish for spawning. So we'd like to protect as much of this eelgrass as possible from being replaced by hydrilla. And so this study and the science here, they were studying bald eagles were dying, and so they did a study and were able to discover that they were eating a lot of cyanobacteria that was in the prey they were consuming, and that was linked to hydrilla in the environment. So just the connection there that's been studied. So I just wanted to mention that. So this is just a summary of where we've been, where the Corps has been working in the lower Connecticut River. So I mentioned in 2024, there were 5 demonstration sites, which had successful demonstrations of the use of herbicides to control hydrilla. We had plans to increase that number to 12 new sites this summer, but because of the funding constraint with the continuing resolution, that's going to be scaled back considerably. But we know now, and we are able to start a new demonstration site in Massachusetts, and we'll show you where that is. And start doing some of the preliminary field work, which is this year would be a dye study, and we'll explain more about what that is and why it's being done. But that's the plan in Massachusetts for this summer. So this is the demonstration site that's kind of drawn in this red rosy color on the left side of the river, right in front of Six Flags. And that's what we're calling the treatment area. It's about 16.9 acres. And this is where we would be focusing on the summer, and it's on the Agawam side of the river. So with that, I will pause and let Kara from DCR give a little additional background about the Massachusetts side of things.
Hi, everybody. This feels like not a lot of people physically in front of me, but that's okay. As Keith said, I am Kara Slawski. I am the Lakes and Ponds Program Manager with DCR. So kind of a little bit of background on hydrilla in Massachusetts as it relates specifically to the Connecticut River. Keith mentioned, hydrilla has been in the Northeast for a long time. It's been in Massachusetts for about 24 or so years. It was first identified on the Cape, but the Monoecious strand that we were showing on the map before. So as it relates to the river specifically, the DCR as an organization has always been involved in the Northeast Aquatic Nuisance Species panel. It's really a panel of state agencies that are organizing, throughout the Northeast to try and coordinate and collaborate on projects and issues that we all face. And so through that, everyone typically has a preference for obviously early detection and rapid response to any new aquatic invasive species, as that's when you have the best chance for success at management. And so, as Keith mentioned, this hydrilla was discovered in the Connecticut River in Massachusetts, probably in that same 2015, 2016 initial time frame. And so all of the states surrounding the Connecticut River were notified of the findings, and Massachusetts began coordinating with New Hampshire and Vermont to survey the river on an annual basis. And so in 2018, DCR hired a vendor, you can go to the next slide, please, to survey the river. And so what they did was they kind of focused on hotspot areas of known plant growth. If you're familiar with the river in Massachusetts at all, there is much less native and invasive plant growth throughout the river than there is in Connecticut. It's much more sandy and rocky. And so this survey focused on those kind of hotspot areas and depths that were appropriate at boat launches, marinas, oxbows, and the tributaries that come in Massachusetts. And so a handful of invasive and non-native species were found, and about twice the amount of native species were observed. And the hydrilla growth in Agawam, where it is now in Six Flags, is about a half an acre in size. And so through that, other areas were surveyed for possible introduction sites, that sort of thing. And DCR began working on a larger scale with neighboring states and the Army Corps and U.S. Fish and Wildlife. So fast forward a little bit to Congamon in Southwick, which was on Keith's map before, as well as one of the sites with the Connecticut River strain of hydrilla. We were notified by the Connecticut Ag Experiment Station staff that they had suspicions that it was also in Congamon. And so they were able to get to Congamon before we were to do a survey and did find hydrilla there. They sent a sample to the Tipperary lab at the University of Wisconsin-Whitewater to be genetically ID'd, just to confirm that it was northern hydrilla and not the other forms of hydrilla that Keith mentioned before. And they did confirm this is the same as is in the river. So we applied for a Fish and Wildlife Rapid Response Early Detection Grant and were awarded that money and sub-awarded that to the town of Southwick so that they could continue managing Congamon's, or begin managing Congamon's hydrilla, as it was the first site outside of the river within Massachusetts that had hydrilla, or this strain of hydrilla. I should have definitely updated this slide. The funding is not currently frozen. This was from a previous presentation when it was a couple months ago. I apologize for that oversight, but as I did mention, to the best of our knowledge right now, this is the only site outside of the river in Massachusetts that does have this Connecticut River strain of hydrilla. So as Keith mentioned, we were curious about what the current extent of hydrilla in the Massachusetts portion of the Connecticut River was. And so based on a survey this fall, which I'll talk about more on the next slide, we knew it was immediately outside of Six Flags in Agawam, and it extends about a quarter of a mile north of there, still on the Agawam side of the river. There is no known growth at all on the eastern shoreline, at least as far as kind of the Connecticut border, where Raspberry Brook and the Fanny Stebbins Wildlife Refuge is. But because of last year's drought, we were unable to really appropriately access that area. So we have some field work cut out for ourselves this year. Next slide, please. So as kind of Keith showed before, the picture on the right was last fall in Agawam. You can see Six Flags in the background, but really had all of that hydrilla growth extending pretty far out into the river, dense topped out growth. And the photo on the left is kind of the density of the hydrilla back when the 2018 survey was done. So certainly a bit more scattered and still provides a little bit of open water in between some of those stems compared to last fall at least. Go ahead. So as I mentioned, we had a full survey of the Connecticut River within Massachusetts done for aquatic invasive species plants last fall. We are appreciative of the warm October weather that we had that allowed us to kind of have a vendor do this without any delays. So first they utilized some satellite imagery to narrow down the areas where plant growth was present in order to be more efficient in their survey methods and areas that they were focusing on. And so they were documenting all aquatic invasive plant observations via an ArcGIS survey project that we have at DCR. So it allowed for some real-time tracking of the survey efforts. And then, depending on what our funding looks like moving forward, we will consider either additional surveys of the tributaries coming into the river or some eDNA work moving forward to kind of save on some labor there. So I know it's a little bit challenging to see because of the base map behind some of these survey points there, but this screenshot is really from the Vermont border to the Connecticut border in Massachusetts. And down at the very bottom, you can see some teal or light blue circles, depending on how your computer displays them. But ultimately, that is the hydrilla growth within Massachusetts. There's no teal elsewhere on that map. I know there's a lot of other colors and circles. For those of you who are wondering, the red is Eurasian water milfoil, the yellow is curly leaf pondweed. There's a few others tucked in there in the Oxbow and Barton's Cove, but not a significant amount of growth overall. Next slide, please.
Thanks, Kara. So I stole this slide from Greg Bugby at the Connecticut Ag Experiment Station. That was a good thing to keep in mind, that the lack of action and or inaction is something to be start to be aware of and wrap your head around with something like hydrilla, because unfortunately that has significant potential negative effects. So we'll just use that moving forward as a reminder. So we'll get into what the management options are and what the Corps is doing with those options. So generally you have these 6 basic tools in the tool belt to try. So I'll read left to right. So the first is EPA research selective herbicides. You can use non-selective biocontrol like grass carp, which is not actually an option in places like Massachusetts. Mechanical harvesting, benthic barriers to block out the sun and try to snuff the plant out, and then also selective biocontrols if they exist. And then those you could always choose to do nothing and the no action plan. So we'll get into next slide. I'll get into more of the background of why herbicides are being studied. So chemical herbicides are the most effective and actually selective way to control hydrilla, which selective means it can actually preserve some plants and just target the hydrilla. So we've been able to work with that. So the Corps has been working with these aquatic herbicides for a while. The, the herbicides, just for everyone's aware, we'll get into more details, but they are heavily regulated. They're readily available. They can treat a very large area pretty quickly. The applications, if done correctly, can actually prevent further spread. So if you can kill the plant before it actually is able to reproduce or form any kind of turions, that would be a really targeted, effective way to treat to prevent additional growth next year and beyond. So that there's a timing component of when they're used that's critical. But the other methods used, like mechanical harvesting, you know, they're very expensive. They're only minimally effective. They can actually cause the plant to spread further, which is an unanticipated result. Or, you know, not why you want to remove it in the first place, but, so yeah, it's a consideration, but we're really targeting the use of these more efficient methods, including herbicides. So again, I'll emphasize that public safety is the top priority for this and using this method. We are only using EPA approved and Massachusetts Department of Agriculture approved herbicides. These products have undergone rigorous testing over many years. The safety is well understood, and the effects on the aquatic environment is all very well understood. So some restrictions or requirements that we adhere to, or for example, like considerations like if in the river, if there is river flooding, you know, no application would occur at that time. And then, you know, we are committed to doing post-treatment monitoring for years after to monitor closely the effects of the hydrilla and also the native plants that are there. But in general, the aquatic herbicides can be used to selectively control invasive plants, and they do not pose significant safety risks. So some information and background about environmental safety when using aquatic herbicides. You know, as soon as they hit the water, it begins to dilute, so the concentrations start to be reduced, and the product begins to degrade immediately. So things like sunlight and presence of microbes, they start to break down the product, and so it breaks it down to a way that there is no residue left behind, so there is no herbicide that persists in the environment with the products that we're investigating. In general, there's no closures to the treated areas, no long-term closures. I think we would just close the area down to allow the contractor to be able to access the area easily and not have anyone in the way. But as soon as they're done, the water is generally deemed safe enough to boat and fish in, or even swim. So, next slide. So for those reasons, you know, we just wanted to make it clear that's the reasoning why herbicides are being selected. It's the best selective control option for the study, is the use of aquatic herbicides. So I will turn over to Dr. Ben Sperry. He's leading up the research team and the research project for this, and he'll walk through more of the details and background on that. So go ahead, Ben.
Thanks, Keith. Hi, everyone. Let's go ahead to the next slide. So, just a little bit of background where I fit in. In addition to being the technical lead for this project, I also lead the Corps' aquatic plant management research team. And essentially we conduct research to help our own agency in operations and finding new management solutions for aquatic invasive species, but also partner with many state and local agencies around the country for those same reasons. But also, we are called in quite often to serve as subject matter experts, not only for aquatic plant control, but some various regulatory pieces related to aquatic herbicides or biological control agents. And we also collaborate with many universities. Keith had a slide up of some of those other research groups that we've worked with on this project. So we're just one piece of that broader R&D network that is focused on this project. Next slide. So reiterating what Keith said on the background of this plant, just probably in different words and a slightly different accent. The Connecticut River is huge, and it's complex. It has a lot of uses, and it has high economic value. So that just the current infested site is a challenge, and broad-reaching system, let alone. The second big challenge in this equation is the genetics of this hydrilla strain. As Keith mentioned, this was new to the U.S., and we're still trying to figure out all of the details about it. But we do know since it invaded the Connecticut River, it rapidly spread and started causing issues with all of the uses of this system. Also, kind of as a setback or another major challenge, unlike many other projects that we get involved in, this one had very minimal applied research conducted until we got the project off the ground. And not the case in Massachusetts, but in Connecticut, there was no agency that was responsible for a publicly funded aquatic plant management program, or more specifically, doing any kind of hydrilla control work. So logic or common sense tells us if we have a problem like hydrilla spreading in these systems, we do need to manage those problems to maintain the uses in local economies that are dependent on it. So that's where we started. Let's go ahead to the next slide. So with a complex issue like this one, we kind of needed a complex research approach. And on the technical side, we use what we call an applied research approach, which is solely focused on applied science rather than basic, with the intended goal to develop environmentally compatible control methods and ultimately provide guidance to those boots on the ground that are going to solve the problem. Unique to this project, though, was because of how new of an issue it was to the United States and the communities and industries that it was affecting at such a rapid pace. We really needed a paired education and outreach program with stakeholders such as yourselves to get everyone up to speed on what we know and the direction that we're going to try to solve this problem. The next couple bullets here are a reminder to folks about setting realistic goals. So in the case of Connecticut River Hydrilla, eradication is not a reality right now, based on resources and just the magnitude of infestation. So we've focused our goals on developing a management program, which takes a lot of time and a lot of effort to set up, with the overarching goal that we need to mitigate the spread of the species to other systems and the rest of the country. And with a successful management program long term, we might be able to bring up that eradication word down the road if we are successful. Next slide. So breaking it down into data gaps, and these are what we've based our research approach on. Firstly is the detailed growth and reproduction of the new hydrilla strain, not only based on conditions in the Connecticut River, but modeled conditions to other systems in the region and the rest of the country, anticipating its spread outside of the current system. Second is site-specific water exchange dynamics in the Connecticut River or infested sites. Now, Massachusetts is lucky in that it does not have a significant tidal influence, but that was a major challenge and continues to be a major challenge in sites in Connecticut. Third is management timing in conjunction with phenological weak points in the life cycle. In other words, understanding when the most optimal time to manage this plant in terms of time of year, so that we are most effective. And then lastly, identifying the most effective and selective chemical control strategies, looking at things like application rates, exposure time requirements, and application techniques, so that we are refining our prescriptions to control the plant, but also maintain any native plant populations in the affected area so that they can recover and repopulate an infested site. Next slide. So I'll break down our research objectives based on those data gaps here. And the first is water exchange and testing herbicide techniques. And this is what we plan to do with Massachusetts DCR in 2025. So in the case of these studies, we use a inert tracer dye called rhodamine WT. It's that red dye you see in the water in that top picture. And this is a fluorescent dye that we can read with specialized sensors, and it moves throughout a water column and with river flow, very similar to how aquatic herbicides move. So it allows us to test the hold time, so to speak, in a potential treatment site, as well as various application approaches or techniques. Think of this as a practice run in a new site so that we understand how the water is moving throughout that site, and then can make a better estimated herbicide prescription whenever the time comes to treat it. Next slide. Secondly is plant phenology, and this is just a technical term for life cycle research. And we did this for 2 years in Connecticut, where we sampled various plots along the river for on a monthly basis to understand where the peak growth events occurred, the environmental factors that drove those growth events to occur, primarily reproductive events, when it's producing these propagules called turions, which type of turions are being produced, and to what extent they're being produced, so that we can then target management prior to those events and eventually wear down the population so it cannot spread further. Next slide. So I wanted to share just some brief results on this part of the research. And this is all based on Connecticut plots. But what we saw is topped out hydrilla in July. So prior to that, you can see on this top left graph here, these plants really start get going in May. You really won't see them visually out on a boat until late June. They'll be right under the surface, and then around that 4th of July time frame, it's really causing a problem. And also you see those graphs starting to go downward in around the September time frame and October. That's just a natural senescence event after the growing season has concluded. So that's really important for us in determining application timing, because we want to control these plants prior to them causing a problem and being at the surface. Next slide. So furthermore out of this work was the reproductive pieces. And these pictures here on your right are the two primary Propagules is the term, or reproductive structures that this plant produces. The first is that axillary turion. These are little buds that grow on the stems in the water column. They also will float downstream. They're attached to the stem and will drop off and infest new sites. The second type is the same structure, but it grows under the sediment and is referred to as a subterranean turion or tuber. And those are problematic as well because they stay put wherever the infestation is. And even though, following management, you can knock out the entire above ground biomass, those are the structures that are going to recruit in that area the following growing season. So did a lot of testing on production of these structures, the timing. We have that pretty well figured out, but I wanted to draw everybody's attention to these numbers that we were seeing in the field on these graphs on the left. So 1,700 turions per meter squared and up to 372 tubers per meter squared. So that's nearly 2,000 or a little over 2,000 actually propagules or potential new plants in a square meter, which is not a very large area, being produced on an annual basis. So as you can imagine, that will accumulate over time and make this problem longer and longer lived in the absence of management. So this is very important to understand how long do we need to manage a site in order to eventually wipe it clean from hydrilla. Next slide. Part 3 is herbicide assays, where we determine what the individual plant relationships are with various herbicide exposure regimes. Not only the target hydrilla strain, but also all of the various non-target native plant species that we want to preserve in these sites. Next slide. So this is very important, and it differs from terrestrial weed control, such as that most people see in their yard or an agricultural setting, because it's not two-dimensional, and we don't just spray and walk away. So we're dealing with 3 dimensions, being underwater and this plant growing in various water depths. Also, we know that water does not stay put in most situations. So the success of a herbicide treatment in aquatics is directly dependent on this relationship here called concentration exposure time. And essentially it's a 2-factor dose response, where the first factor is herbicide concentration, or how much herbicide is being put into the volume of water. And then secondly is the length of time that concentration is in contact with the target plants in that volume of water. And we have to have both to be successful. If we don't get both of these factors just right, we will end up with sublethal plant response or failure to control the plant. And to make matters worse, if we're dealing with aquatic weeds in your typical lake or pond system, those usually don't have a high water exchange rate or a turnover rate. So we have long exposure time potential, and it's much easier to control plants in that situation. However, in flowing water or small plots, partial system treatments where water exchange rates are going to be much higher, it's more challenging, and that's why we need to understand these concepts at a very precise scale in order to be successful. Next slide. So this same concept, like I mentioned, is important for selectivity towards native plants. And I'll use this graph to kind of explain that. In this example, the bands are different concentration exposure times of a various herbicide. Our target here is Eurasian water milfoil. So if a treatment occurs in this blue band where it says Eurasian water milfoil, that will control that plant. However, these other bands that say coontail and spatterdock, those are native species that we would want to not impact there. And those are typically controlled at higher concentrations and longer exposure times. So what we're doing at each site in this project is determining that water exchange and then identifying a herbicide prescription so that we can stay in a blue band here where we are only the rate is low enough to where we are only affecting that invasive species long term. Next slide. So moving to step 4 in the process this is where we were at in Connecticut last year. And this is where we put all of our smaller scale study information together. And we call this field verification studies or treatment demonstrations. So all of the herbicide concentration exposure time data, the phenology or life cycle research, and the water exchange rate research, that all goes in to form a prescription for a various site. We usually start at a small operational scale and conduct a commercial aquatic herbicide treatment. However, we don't just do the treatment and walk away. We are very detailed in the amount of data that we collect post-treatment. And in that process, we're measuring things like the herbicide concentration over time throughout the plot, various ways of measuring plant control and non-target plant response over at least a year in most cases to understand what is the restoration process looking like initially after these treatments. Next slide. And then part 5 of the research, this is kind of more on the basic side that's going on in the background, but it doesn't necessarily pay off immediately for field work. But this is where we uncover some clues from time to time that makes the operational components much easier. I can really just boil it down to biology and ecology here. So what we've been doing with this new strain, for example, is determining if it can cross with other hydrilla strains, where we are artificially pollinating other dioecious and monoecious hydrillas with Connecticut River hydrilla pollen to see if seed production is occurring, which we found out. But luckily, we haven't seen the flowers co-occur in terms of environmental requirements. So I'm crossing my fingers that that's not a potential in the field. We're also looking at things like turion sprouting requirements. As this plant may be moving further north or south in the country, we need to know what time of year or what potential environments can it capitalize in and outcompete native plants in new systems. Next slide. And then lastly is our end user guidance. So this goes back to our initial goal with applied research is we do all of this to eventually get the most up-to-date and best science available out to the management agencies to go out and solve the problem. Now this takes form of master guidance documents, resource management workshops, and management protocols for various entities. And we connect with those entities doing the work to understand also where new issues pop up so that we can use that as a feedback loop back to the research to further improve those management techniques. Next slide. So real briefly, I'll go through kind of the early results of some of our treatments last year in Connecticut. We had these 5 representative sites, used various herbicide prescriptions, and as you can see here, they vary in scale and rates and timings. A lot of that has to do with logistics as well as tidal timings. But anyways, I'll get into it. Next slide. The first site here is Keeney Cove. And as you can see, it's kind of a pond with a road that goes through it that the water flow is somewhat restricted and eventually connects down to the main river. What you're looking at here is satellite imagery of pre and post treatment with a herbicide called fluoroperoxifin benzoyl. So on the B panel, this is pre-treatment, and those red dots signify our survey detections of hydrilla. And the redder the dot, the denser the hydrilla, and that'll be the same theme moving forward through these sites. So pre-treatment, as you can see, a lot of dense hydrilla throughout that section of the site, and then in the C panel, what we're looking at is 2 months post-treatment. And as you can see, most of those dots disappear, and all of them became a lighter shade of red, with only 2 detections occurring in those corner pockets. So this is a good example of we put all the pieces together, and we're probably up around 95% effective with the treatment. But we can't walk away just yet because we need to know what happened at those 2 detection sites post-treatment, which is probably going to take some more water exchange studies and refinement of that herbicide prescription. Next slide. So just controlling hydrilla isn't all of the story. So we need to know what else happened with the other plant species in there. So this is what we call relative abundance data throughout the site. As you can see, starting with hydrilla on the left, massive reduction in hydrilla post-treatment from July to September. Also, there was a lot of water chestnut in this site, and we were somewhat surprised that it also was decreased. It's another invasive species that floats on the surface, so that was a good bonus. Coontail was a little bit of a surprise. That's a native species, and as you can see, it was at pretty low abundance throughout the plot, likely due to being outcompeted by the invasives, and also was decreased a bit. So that's something we're going to look at in the future to document its recovery post-treatment. And then duckweed, another somewhat desirable species in some cases, increased after treatment, which is attributed to increased water flow and surface fetch in that area once those invasives are dropped out. Next slide. So just a visual representation here of this site. On the left picture, this is pre-treatment. That's all water chestnut, that other invasive species I mentioned in one of the corners of the site. And as you can see on the right-hand side, that's about 3 months post-treatment, where all of that site became usable again and really opened up the water surface for all of the wildlife there. So unexpected result. We weren't really expecting to control water chestnut, but that's why we follow these treatments, to see what other information we can collect. Next slide. Excuse me. Our next site here is Chapman Pond. We use the same herbicide, and again, we are looking at that pre and post-treatment satellite imagery. As you can see, pre-treatment, we had a ring of hydrilla around the littoral zone on the shoreline at pretty high densities overall. And again, post-treatment, probably 90-95% effective there, with just a couple anomalies that we need to figure out whether I think these two, those 2 sites, the pink dots, those are actually probably from drains where we got some rainfall or new untreated water flushed the herbicide out of those spots. But regardless, pretty effective on the hydrilla control front. Next slide. In terms of other species here, I'll skip hydrilla, go to coontail. Well, we saw a little bit of injury with coontail, but not completely wiped out. So we'll be watching it and anticipating recovery this spring. Eelgrass, or Vallisneria americana, a very desirable native plant. It's pretty wide known that this herbicide does not affect it. So as you can see there by those bars, was not increased or decreased. Elodea canadensis, we saw some sensitivity of that plant. That is a native, so it's another one we'll be tracking. EWM stands for Eurasian water milfoil, another invasive and kind of an added benefit of this treatment. And then pickerel weed there. That's a shoreline weed. We saw some injury with it from this herbicide, and we'll be tracking its recovery in the spring after it gets a chance to warm up. Next slide. Moving on to the next slide, our site is Chester Boat Basin. So new treatment approach here. This was treated with 2 herbicides in combination called Indithol and Diquat. They're a little bit faster acting, and as you can see in that B panel, very thick hydrilla in high abundance throughout this marina. And post treatment, probably at 85% control. With a few low density detections there. We're not real sure if those were misses in terms of weed escapes after treatment, or if they were introduced fragments from the main river because of the high cycling in this site. Next slide. So again, in addition to the hydrilla control, we actually got quite a bit of other plant species response data here from this. So coontail reductions, we anticipated that. Kabamba, another invasive, no response. It actually increased in area after the hydrilla competition was removed. Southern naiad and Elodea canadensis also were reduced. No surprise there, but those species generally come back on an annual basis. And then on the emergent side, we had pickerel weed and wild rice with no effect of the herbicide treatment, and also saw a slight increase in eelgrass abundance following treatment with Indithol and Diquat. Next slide. So I think this is my last one I'll present. This is Selden Cove, which is kind of an oxbow off of a creek that's connected with the main river. Fairly fast water exchange, shallow water, and very dense hydrilla, as you can see in that B panel pre-treatment. And post-treatment, wiped a lot of it out, and I still need to update this slide. Most of those detections were actually Elodea canadensis, a native. So we'll see that on the next graph. So go ahead and flip the slide for me. So as you could kind of see from the maps there, we had very good reductions in hydrilla abundance. We also got some water chestnut control out of this site. Coontail was actually controlled unintentionally here, but Elodea canadensis, this was the most selective treatment that we've seen towards that species, which is a desirable native. Also, eelgrass, no effect, and probably statistically no effect with pickerel weed and wild rice, even though it looks like an increase on this graph. So, very selective treatment in Selden Cove. And next slide, I think this goes back to Keith.
Yep. Thank you, Ben. So yeah, we're almost to the end here, but I just wanted to go through just one of the next steps after doing this in the field. Use of application of herbicides is the post-treatment monitoring. And so this is a lot of the information we collect with that process. So we really want to track the native plant response, like Ben was walking everyone through, with the results and how hydrilla is doing. But we monitor closely herbicide concentrations over time, track if there's any threatened endangered plant species in the area. We specifically do surveys for those to see how they're doing. But we are really looking to do these surveys for up to 2 years, potentially up to 5, depending on the site conditions and needs. But just want to make that clear that, we're not just treating these areas and walking away. This really is part of a research project, and we're monitoring the results and the efforts very closely. So for, we printed some fact sheets out. This is for the folks online. These are printed out on easels in the room. It's the same information, but these are all available online to download as PDFs from the website. But just wanted to flag that for everyone. We do have resources available to help understand a lot of the information that was presented here. this evening. And so I have links for the website coming up, but I'll just reiterate that the timeline for this summer is to do a dye study in Agawam with the eye, looking forward with the plan to potentially do an herbicide treatment in that spot next following summer in 2026. But again, we'll just caveat that with, there's potential constraints, whether it be funding or permitting. We're gonna do what we can to be ready to take advantage of that timeline and carry out a treatment there as part of the research project. So that's our general plan right now. And so that's the end. So thank you very much. And if you advance one more, there's the links for the project website, and there's an online ArcGIS story map that has a little more user interactive imagery and videos and resources to help folks understand the research project details. And then there's an email address here. If you send an email there and would like to get in touch with us, that goes to me and Dr. Ben Sperry directly. So feel free to use that email address to get in touch with us with any questions. But with that, we can conclude the slide presentation and open it up for questions and dialogue. So any coming in online there? Most of the attendees are online. So.
I don't have any questions. Oh, Kelly Bierman has a question.
Hi, can you hear me okay?
Yes.
Hi, thanks for the information. It's really useful. Kelly Bierman from Connecticut River Conservancy. I'm the new Aquatic Invasive Species Program Manager. I just had a question about Keeney Cove specifically and the data around water chestnut and those results. I'm just wondering if and how those results account for the hand pulling done last year. Because I know quite a bit of hand pulling was done on water chestnut and Keeney Cove, so I'm just curious about that.
Hey, Kelly. Yeah, so unfortunately, the hand pulling started before we did that treatment. However, anecdotally, I can tell you I've watched the crews kind of go backwards in terms of area. That site was very infested, and it seemed like it was outgrowing any of the physical removal efforts. So it would that in combination with the herbicide treatment would be hard to put any hard numbers to the hand pulling efforts distinctly.
I don't have any other questions in the chat. Oh, Lou Cornett.
Yes, good evening. Recreational user of the Connecticut River in Massachusetts. I'm representing one of the marinas based in that area. What can we do? This is really disturbing news watching this presentation. I don't think the average person here in this area has any idea about the magnitude of the problem, and it seems pretty gloomy. So what can we do as individuals reaching out to our legislators? Do you have any recommended action plans for us?
Yeah, I can. Hi, Lou. I'm Kara, as I addressed earlier, but definitely tell everyone that you know, the more people that are aware of this and can support this and be involved and interested, the better ultimately, because we want everybody to be aware of what's going on and the work that the Corps is doing in partnership with us at DCR. And public education and outreach is such a big portion of all work that the Corps does or we do at DCR. And ultimately, that's why we're having these types of public meetings. So people like yourself and others can learn more. And yes, you are correct. Keith's slides are definitely disturbing in some points and the photos are not great. And those are not meant to scare anyone in this presentation, but truly just to show you what could happen to the Massachusetts part of the river if we continue to let the hydrilla expand and grow here. So like Keith said, they have great resources on the project website that the Corps has, but in addition to that, if you wanted to reach out to us at DCR, you can definitely do that as well, because we can provide you kind of general aquatic invasive species information, as well as signage that you can put up at your boat ramps and other marinas that you know, are friendly with, that sort of thing. You know, we're happy to help in that sort of outreach portion of it as well. And though our website is not on Keith's slide here, if you just Google DCR Lakes and Ponds, or, you know, mass.gov, I think / lakes and ponds, you will find us. We have a generic email address on there. You can reach out, and we can coordinate with you. We all, you know, cover the whole state. We're a small but mighty team, but we're happy to spread the wealth and share signs and booklets and handouts. We teach plant ID classes. We do the whole shebang. So whatever you need, we're here to support you.
Well, thank you very much. I just received an email today from an organization called Friends of the Franny Stebbins Reservation, which is located right near our marina. And they were talking about their efforts on not this particular invasive plant, but several others. So this seems to be a oft occurring trend nowadays. Is this all based on climate warming? Why are we seeing so much all of a sudden?
I think the climate warming is certainly not helping the cause. As Keith mentioned, you know, he showed those climate projection maps from the Corps that show potential areas for hydrilla growth and infestation moving forward. So that is definitely one piece of it. The other piece too is that people and wildlife are continuing to travel and recreate in water bodies that are infested. I would say more water bodies in Massachusetts are infested with at least one aquatic invasive plant, if not multiple, as opposed to water bodies that have none. You know, I can only think of a few, maybe 5 off the top of my head, that have no aquatic invasive plants. Granted, there's probably many more out there. Massachusetts has about 3,500 water bodies. But they're definitely out there, and unfortunately, if you're not aware of what you're seeing in the water, it's much more out of sight, out of mind than when you see a terrestrial invasive. You know, you start seeing bittersweet growing somewhere, or Japanese knotweed, or Phragmites, or even poison ivy. And once you see it somewhere, you can't unsee it somewhere else. You know, you are always aware of its presence. And unfortunately, Lou, I will tell you that once you see this hydrilla and or, you know, some of the other invasives that are in the river, you'll never be somewhere in the river and probably not notice them now. So.
Well, thank you very much for that information. Very informative. And I will take it back to the other recreational users of this portion of the Connecticut River. Thank you.
And Lou, I'll just also say that we are hosting the Corps as a quarterly meeting with marina owners and operators to give updates and to field questions directly from you. So if I haven't added you to that invite yet, I haven't had a chance to sit down, but that'll be, I'll look for folks like yourself to add to that, to invite you to those meetings.
Great. I did receive that in the information. Look forward to working with you, and there'll be a lot of other people as well.
Great. And I should say, I should have updated this slide to add, there is a Massachusetts specific website for the project that has Kara and DCR's contact information on that as well. So that was my fault. We just did this presentation in Connecticut last week, and I didn't think to update that link for Massachusetts. But that is available. And hopefully, if you Google something like Corps of Engineers hydrilla in Massachusetts, you may actually just get it that way too. So, any other questions?
Timothy Timmerman.
Hi there. I'm Tim Timmerman from the EPA out of our Boston office. And I want to thank you for, even though you don't have a lot of people in the room with you, know that the presentation is very well received. And I have only caught bits and pieces of the presentations in the past that everybody from our offices attended and told me I should try to catch one on it. So I'm glad I did tonight. I'm glad to get the update. Sorry to hear about your funding. Hopefully that'll get resolved. I think it's important work. It's really interesting. It's interesting, the unexpected results slide. I think that one's the one that caught my eye when I was looking through it in past presentations. But one really wonky, just curiosity. Is there any differences between the Commonwealth of Massachusetts and the state of Connecticut with respect to any dramatic differences you've noticed in the regulatory environment for getting, permissions to do this? And it's something that I don't know about, so I was just curious.
Yeah, I think we're aware of some differences. We I should say we haven't gone through that process in Massachusetts yet. We're working very closely with DCR and Kara's office. We've gone through that now a couple of years in Connecticut, but we do expect it to be different. They're not going to be the same experience, but yeah, I think not much more to say other than there are differences, and we're just working through those. But I don't know if we want to get any more specifics.
No, you don't. That's good enough. I just didn't know if there was any contrast that you saw, or no, just really wanted to thank you and know that people are out there listening, and yeah, it's one of those things. I think you can't unsee it, right? Once you learn about it, then you tell your friends. So that's you build the momentum that way.
That's right. Thanks, Tim.
And I have a comment. From Lauren Cruz. She says, thanks for the presentation. She's in the Northeast Invasive Species Coordinators.
Sorry, I realized that was a very long comment after I hit enter. I just want to say thank you. Sorry I couldn't be there in person, but I'm the invasive species coordinator for the Northeast region for National Wildlife Refuges. I know you all got this through Dave, and I was going to alert the rest of Fanny Stebbins staff of the great work you're doing. But if you're ever working by other National Wildlife Refuges, feel free to reach out to me and I'm happy to help.
Thank you so much. We'll definitely be reaching out probably more about Fanny Stebbins to either you or Dave, because now that hopefully the river is a little bit higher than it was last fall amid the severe drought, we definitely want to get back in there and see how far up into Raspberry Brook we can access with a boat, because there is definitely hydrilla there where kind of that Cove Peninsula meets the main part of the river right at the Mass Connecticut border. But we were unable to access it just because of the low water levels last fall.
Gotcha. Yeah, happy to help however. Feel free to contact us both then.
Perfect. Thank you.
Yeah, thank you.
I don't have any other questions. Oh, one more?
In the room, yes.
I guess one question, how many people are online watching this?
9 people.
There were a few that dropped off, I saw.
I know
How many are you?
Sorry?
How many people watching?
There's 9 right now.
Okay.
Yeah.
I'm looking at this chart and I do have some background in science. This is very difficult. Okay. How do we, on conservation agro, how do we approach this? I mean, I think somewhere along the line, we should tell the council in a public hearing that this is a problem. But, the problem is you're talking about a number of different invasive species. How many of those in that part of the Connecticut River? Or should we just concentrate on the one that's a problem at this point?
I mean, they all have their concerning elements, but I think we're all a little shocked at how aggressive this Connecticut River strain of hydrilla specifically grows. I think people have never seen anything like it. So that's kind of the priority right now, at least for the Corps. But yeah, I think there's Dr. Ben Sperry's, his program, they work on all aquatic invasive species in general. So
There's your treatment's coming up pretty soon, according to this letter. Being done in June and now we're already almost in May.
Yeah, so in Massachusetts, we would be doing a dye study only, it's not an herbicide treatment, yeah. Yeah, so that would inform the future, herbicide treatment. But, you know, compared to Connecticut, you know, Hydrilla hasn't done what it, in this state, what it has done in Connecticut, so we want to be mindful and stay ahead of it in Massachusetts, so we're glad that we have that
I'm just
advantage.
thinking, how's the
Yeah.
best way? I think if we probably said something to the council, then had a public hearing on it, or just an informal hearing on it, it might work something like that.
Sure,
Yeah, we can definitely, we certainly have had, conversations with your agent
I
about.
know that, I'm aware of that, that's why I'm here tonight.
Perfect. So yeah, we can definitely schedule something. Absolutely. It's not a problem at all.
Talk to Karen on that, how we approach this.
yeah, we're happy to coordinate with you, we want to work with the local towns and entities as much as possible. It is a challenge to get the word out, you know, there's a lot of folks were invited.
Getting the word out is that's why I asked the question of how
Yep.
many people.
Yep.
Yeah.
Because we have 28,000 people in Agawam, how many are gonna be concerned, and how do we get them the information?
Right, and we did reach out to a number of media outlets, and one group said they would come and actually they were interested in filming this, but, you know, obviously not here. So yeah, it's a challenge to get the word out, but I think we're just forming new relationships in Massachusetts, so I appreciate your concern and we'll more than be happy to work with you on how to figure out how to get the word out.
I'm looking at, so I'm looking at the charts and I think we'd have to inform the people that we have a problem, and probably the best way would be doing it, just putting it, we have this problem with this invasive species, it's hit Agawam, it's a problem. And then we can hold maybe I wouldn't do an hour and a half or 2 hours, but figuring out how the best way to
Yeah.
approach this to people, I'm not sure,
Yep. No, absolutely. I think we wanted to maximize a lot of the backgrounds, but we don't need to do that again in the future. This is recorded. We can post this meeting so that I'll be there in the future. But it was important to kind of do that in a new area. Yeah.
We should do it pretty soon.
Yeah,
We have
I
one
think
more.
we'd be doing a determination on the die, or somebody said, for conservation.
Yes, so we are on the fence about that because we've been given kind of mixed guidance from both conservation and DEP, as it, it doesn't necessarily require a permit, as dye is not
I
an
don't
herbicide.
have a problem.
So, yeah, we're still working through that with the Corps. And, hopefully we'll have some answers for everybody soon as it relates to kind of a more specific timeline for the dye.
I'm more concerned to get, even if we have determination, as we get the thing out, that we have a problem, make the public aware of it, of some sort of maybe a half hour or something, so people are interested that something's gonna happen to the river, be aware.
Yeah, absolutely.
yeah, we definitely want to be mindful of that and for sure.
Yeah. But yeah, we can, even just schedule a meeting, the three of us and Taryn, and, kind of brainstorm a little bit on
Yeah.
how maybe best to share the.
Well, that's three, probably from conservation, not four. Thank you.
Yeah, you're very welcome.
We have a question from the shoestring.
Hi, can you hear me?
Yes,
Hi, my name is Shelby Lee, and I'm with the Shoestring. We're a Western Mass news outlet. On one of the slides, I did see that there was like anticipated post-treatment monitoring, and then at one point there was a 1 to 5 years mentioned, and then I think Benjamin had said that was a year of monitoring. So there's just kind of a variation in that number. I was hoping you could address for the monitoring post-treatment if you get to the treatment phase, And also, is that monitoring window, is that somewhat influenced potentially by funding availability? Is that kind of what the variability there is?
I think, Ben,
Yeah,
Ben,
this has been, yeah. So what we do, especially for northern sites that have a heavy winter influence, is shoot for at least 1 year post treatment. However, some of these sites, in a perfect world where funding is coming through, they'll have several treatment iterations. So we're gonna constantly be on that 1 year cycle post treatment. Does that kind of make sense?
Yes, thank you. And then I had another follow-up question if there's time and not other people waiting, just about kind of the herbicide treatment in general.
Go for it.
Cool. You had mentioned kind of figuring out like the appropriate dosage and time exposure as you're studying this plant species. Is there a potential for developing like herbicide resistance to like a exposure to the herbicide that doesn't completely, deal with the group you're targeting or the area you're treating? Have you seen that in your studies, or has that not not been a long enough time period in the studies for really answer for that question?
So great question. There is herbicide resistance in hydrilla in Florida, but that was a result of probably a decade of over reliance on one single herbicide over and over and over again. So lesson learned. That's why we typically, when operations get off the ground, they rotate different herbicide active ingredients or use combinations. We kind of, all those practices, we use the term herbicide resistance mitigation practices. So that is being practiced in this study or demonstration project, but we've literally only treated those 5 sites one time. So we're not far enough along to demonstrate that, yes, but it is under consideration for long-term planning.
And this might be kind of a common sense question, but is there an impact to non-plant species in the river from herbicide use that you've observed at all? Like I know mussels and stuff breed in the Connecticut River. Have we seen any accumulation in non-plant species of those materials? Or again, is it kind of early in studies to know much about that?
Another great question. So that is why we exclusively use US EPA registered products, which have gone through rigorous testing and have to meet a certain risk criteria to not bioaccumulate or biomagnify in the environment. And there's an abundance of non-plant, non-target organism toxicology data that informs those registrations. So these products that are registered for aquatic use are some of the cleanest chemistry in terms of all herbicides available, which actually makes it quite challenging to get them to work sometimes because they are so short-lived in the environment.
Thank you for the question and answer in period, and also the slides were very helpful. Thanks.
you.
Okay. That's everybody.
Okay. Well, I think we'll move to end this meeting now, but doesn't mean we don't want to hear from folks in the future. We'll continue to coordinate and get messaging out and be in touch about the actions that the Corps is doing, and when we have updates and more results from the testing, that'll all become made public. So we hope to just keep spreading the word, and I'm glad everyone online was able to attend as well. So we'll put this recording up on our website. I think this was a successful recording of the meeting to get some of the information out and some of the great questions. So yeah, I think we'll end it here. Thank you so much.